this whole long episode on cellular choreography. I'm very thrilled to have someone who is actually interested in regenerative medicine, stem cell biology, is a young scientist, Basudha Banerjee. Basudha, it's really great to have you here.
Thank you, Dr. Khurana.
So let's do a tour of the cell, not in isolation like most biologists do, but through the lens of regeneration, and think of it as a guided journey through the organelles that breathe life into our therapeutics. Let's start at the core, the nucleus, our genetic control hub. In stem cell therapy, it's not just a storage facility of DNA, it's active decision maker.
Absolutely. The chromatin architecture in the nucleus, it determines whether a stem cell remains pluripotent or whether it commits to a lineage. Epigenetic reprogramming, say via the Yamanaka factors, they rely on making the chromatin more accessible. So think of chromatin as the script and transcription factors, they are the actors waiting for their cues.
And the nucleolus, underrated but vital. Without efficient ribosome biogenesis, protein synthesis grinds to a halt. Regenerative processes demand high translational throughput. So nucleolar activity is actually a proxy for regenerative capacity.
So, with that, let us discuss something about the main protein factory of the cell. So, let's discuss about more of ribosomes, the endoplasmic reticulum, and Golgi apparatus. Once the script is read, it is the showtime for the ribosomes. Free ribosomes produce cytoplasmic proteins, while those on the rough endoplasmic reticulum handle secretory and membrane bound proteins.
And we can't forget the smooth endoplasmic reticulum, especially in hepatic regenerative models. It's central for lipid biosynthesis and xenobiotic detoxification, which are key in stem cell derived liver organoids.
Exactly. And then comes the Golgi apparatus, which steps into fine tune, package, and sort. It is like the final quality control checkpoint. In a lab's engineered secretory cells, fine tuning the Golgi function literally determines whether a protein is bioactive or whether it is immunogenic.
Yep. So now let's shift gears to mitochondria. Let's zoom into mitochondria, which are my favorite, at least for now. My own research team at IonCure is very interested in it. When I was in a place in Lucknow, India, we were doing some work from home projects on mitochondrial bypasses, which we didn't complete then. Various challenges of citizen science and Indian bureaucratic science, but we had several initial leads in silico work. So this on neurodegenerative diseases, cancers, and the cancer story goes back to even some interesting old interaction with Jim Watson. So mitochondria have been my favorite for last around 14, 15 years. So that leaving my personal bias aside, they're not just powerhouses, but decision makers. In stem cells, a shift from glycolysis to oxidative phosphorylation is a cue for differentiation.
And in addition to this, we also have the mitochondrial dynamics, say the fusion, fission, and mitophagy. These all are very critical. Aging cells often accumulate damaged mitochondria, and this impairs the entire regenerative response. So, targeting mitophagy pathways with compounds like urolithin A, as we previously discussed, or even NAD plus boosters, can rejuvenate the stem cells.
Yes, mitochondrial heteroplasmy and mitochondrial DNA mutations also impact outcomes in iPSC-derived therapies. AI can predict impact of mitochondrial variants in patient-derived organoids. AI is making huge impact. Most of it is currently at the lab bench space. It's not yet reached your pharma, but we hope from lab bench to bedside, it can be a whole revolution out there where AI is actually telling you which mitochondrial variants are going to make huge impact in regenerative medicine. So it's what would have been science fiction 20 years ago is becoming reality now.
So this is pretty interesting, and we are very hopeful about the integration of AI with all the healthcare and the lab experiments. And with this, let us dive into the lysosomes and peroxisomes that basically are known for the detoxification. So lysosome is generally the cell recycler. In neurodegenerative disorders, impaired lysosomal clearance leads to toxic buildup. So, enhancing lysosomal function has become a regeneration strategy in Parkinson's and ALS models.
Yeah, and the peroxisomes are often overlooked. Their role in fatty acid oxidation and ROS detoxification becomes crucial in cardiac stem cell therapy. If we don't maintain redox homeostasis, we end up promoting fibrosis instead of regeneration.
Absolutely. So, with that, let us discuss something about the cytoplasm, cytoskeleton in general, the basic infrastructure. So, cytoplasm isn't just a filler, it's a dynamic matrix where all these conversations happen. Metabolites, mRNA, or even the signaling complexes, they all interact here.
Then there is cytoskeleton, microtubules, intermediate filaments, actin. Without proper cytoskeletal organization, stem cell migration and tissue integration fail.
And then we also cannot forget the centrioles. They are the main architects of cell division. And in regenerative context, like skin renewal or gut epithelium repair, precise mitotic control is very vital.
Yeah, lastly, plasma membrane, it's a gatekeeper. Its lipid composition, receptor density, and mechanotransduction properties dictate how stem cells sense their environment. Actually, I'll tell you something which is at an idea level. It may be more crazy as of now.
Mhm.
We did some interesting thing. We analyzed from existing data, so it's not primary data. It's secondary data from very good, amazing groups from UCSD, John Hopkins, so very prestigious groups. So we looked at data, we looked at the quality of labs, track record, whether we can trust it, on what changes happen in cell membrane with aging or with diseases.
Hmm.
And then we just simply said, can we make edible oil as a way to make people age slower? So what things do elderly need, which is different? So instead of one cooking oil for everyone,
Mhm.
can in a future, at least for those who can afford, maybe in developed societies for everyone, maybe eventually all of humanity, can you make things which are very personalized? Or at least can you make things which are for categories? Someone's going through a certain kind of cancer. If their food is made in a certain slight change to fatty acid composition, like certain oils are different, will that make a small difference, 2-3% difference to their health? And obviously, which includes a lot of cooking, other things. So this is very early days. A lot of good ideas do fail, right? So it's at that idea level. We did a lot of analysis, and turns out that the guy who promised to do a lot of things just vanished once he got recommendation letter. That has been the story of citizen science in India.
Mhm.
So, so the project got abandoned, and then I met even 2, 3 interesting aspiring entrepreneurs who wanted to pick, but their ability to struggle beyond 2, 3 months was limited. So initially was very optimistic about a guy because he flew in from Kolkata just to meet me about this project. He had done PhD in chemistry from Long Island, where different institute, but I was at Long Island, Cold Spring Harbor. So we were trying to see if there is some common ground.
Mhm.
But he, I think, just realized that this is how many millions of dollars and how many years of struggle is required. I think he just gave up. He was thinking it's an easy thing. But yeah, this was an interesting idea on which no one else is even doing. This is a 7-year-old story. So I'm surprised that in 7 years, people haven't really picked up on it. I wanted to create a regenerative medicine company based on sort of designer fat, designer fats as you age, right? So basically, take people, see if we can do it. Obviously, this is a long shot, right? From what is
Mhm.
deficient, just simply if you add it, how does it get broken up? Whether that'll actually contribute to cell membrane or non-membrane fatty acid requirement. There's a lot of steps involved. It's not as simple that something goes down, you just increase this. In fact, some of our analysis even suggested that you increase certain other things
Mm.
may make the plasma membrane healthier.
Okay.
But that was an interesting start. It can still be a huge thing. And um, I think something which is very interesting that people ignore plasma membrane, right? So everyone looks at one or two things, including mitochondria, which is my favorite. But there are, I think, what AI can give us in future. Sorry for a tangent. You, Basudha, know I like sort of this is my habit.
Mhm.
Maybe you can say still, despite being an entrepreneur, still haven't completely shed off my lost professor attitude fully. But the thing is that where I would like to drive home an important point to our listeners is that instead of just thinking of one thing as a fix, the future belongs to AI, which integrates all approaches, every single organelle, then looking at tissue level, looking at organ level, right? Think of it as if your car is getting old. You just don't say, I'm going to just work on carburetor and I'll ignore everything else. Unfortunately, 99% biologists operate that way. I know how to fix air filter or I know how to fix carburetor and I'm only going to work on that and you'll be all right. In fact, it's not just research scientists who have this myopia. Rather sad stories of my friends, in fact, co-founders in some businesses going through, like one of my friends, I'll not mention name for the privacy, Basudha, you know probably who I'm talking about.
Mm.
His father is going through uh, multiple myeloma. So there are cancer things, there is lung issue, there is heart issue, which has been lingering for various time, and now there are various kidney issues.
Mm.
So the different doctors don't really talk to each other.
Yeah.
So you have to convey a lot of information. So medical system works in isolation, science works in isolation.
Yeah.
Why the future is AI
Mm.
is to take every component as we age, as we have disease,
Mm.
and to put it together. And not just approach things from medicine point of view, approach from medicine, from even some potentially might sound science fiction, some slightly anti-aging vaccination, like things which will slow down certain things. Very controversial RNA therapeutics. The future belongs to obviously a lot of nanotechnology. The future belongs to a lot of brain chips to
Mm.
keep us active in the declining brain health times. It belongs to targeting every organelle. It belongs to cleaning up within cell and within tissue. Longevity is definitely possible. I'm not sure if I subscribe to Brian Johnson. I'm not sure if immortality is possible. But I think in our lifetime, maybe your lifetime, you're probably 20 years or 25 years younger than me. In my lifetime, maybe 20-30% chance that we can push the lifetime to 120, 130 years. Probably in your case, 70-80% chance that that can be done. But I think it'll take a very integrative approach for AI to do that. So essentially, that's the thing. Now let's get back to from my tangent, which went on plasma membrane.
Mm.
Let's return to something because we have still left out several things like vesicles and exosomes. What do you say, Vasudha?
Absolutely. So I would just first like to appreciate that oil thing that you were working on for the cancer, or rather even a lot of other diseases. That seems very insightful. And of course, there is a lot of time and effort needed to have great success on that. And yes, Dr. Khurana, let's get back to the discussion of vesicles and junctions, basically trafficking, vesicle trafficking, or trafficking anything between different organelles. So vesicles are critical for all the intracellular logistics, especially the exosomes in regenerative medicine. They carry microRNAs, proteins, and even mitochondria to the damaged tissues.
Yeah, and cellular junctions, desmosomes for Edison, gap junctions for electrical coupling, especially in cardiac regeneration. Without these engineered tissues fall apart literally.
And also, we have the hemidesmosomes. They play a crucial role in epithelial integrity, like all the skin grafts. And without proper anchorage, you see all those graft rejection and blistering issues rising up.
Yeah, and we have even other things like specialized organelles, niche structures. Let's talk of melanosomes. Not just pigmentation, they've shown interesting ROS buffering capacity in certain regenerative skin models. I'll not go again into a very long tangent. Basudha, you and I can talk offline about certain pigment synthesis and certain pathway. And I was wrongly barking on a wrong tree with a postdoc. Again in Lucknow, I paid 2 years salary beyond institutional funding, some even from my personal money to someone, which took me few days to reconstruct, but I couldn't get the lady to build that whole pathway. And but there were very interesting insights. And again, something I'm surprised hasn't fully been appreciated in Parkinson's. There is off on and off some literature which has been there for a long time because, as you know, Parkinson's affects neurons in substantia nigra, right? Sort of pars compacta. It's dopaminergic neurons in substantia nigra pars compacta. So these were called substantia nigra because they were dark. They have pigments, and we realized pigments are not just it's not just pigmented neurons. They have something specifically to do with ROS and in fact why things die soon and why they need to be protected. Longer story, maybe we can create a separate discussion on it, but I would drive home a point on melanosomes in general, that they are not just for pigmentation. They show very interesting ROS buffering capacity in certain regenerative skin models, and as I just alluded to it, that pigments may play a role beyond skin models, but in fact even in brain. Over to you, Vasudha.
Yes, Dr. Khurana. So, addition to this, I would also like to add about the lipid droplets because they are the key in metabolic flexibility. Say for the adipose tissue derived stem cells, they serve as both fuel reserves as well as signaling hubs.
And photoreceptor discs, myofibrils, neurofibrils, each specialized for specific tissues function. We can recapitulate these in organoids now, and possibilities are immense as things are unfolding.
And then we also have synaptic vesicles, especially in neuronal regeneration. It is not just about growing axons, but it is more about restoring the functional connectivity.
Absolutely. I'll not. There is a force of 7 horses preventing me from taking a tangent on synaptic vesicles and growth. It's one of the stories of my PhD. I did 7 different small projects, some major, some minor. Unfortunately, published only 2 from my PhD. And my PhD boss later, when I was at Lucknow, almost 10 years after PhD, was suggesting that I can publish them on my own. Earlier, he was. I have some recordings of him basically being concerned that I'll compete with him and outcompete. So he, Americans can be like Indians, also petty. So, but how synaptic vesicles play into regeneration in the context of serotonin is something I personally spent years on. So that's a separate tangent for some other time. Now let's try to close all of this, how organelles come together. Let's think of organelle synergy in regeneration. So Basudha, if we were to summarize, over to you.
I would say it is all about organelle synergy. Regeneration is not just genes being turned on, but it is more like a cellular ballet and orchestrated across membranes, vesicles, filaments, and enzymes.
And the future AI guided organelle engineering, nanorobotic cargo delivery, real time cellular feedback systems.
Where we don't just fix the tissues, rather, we focus on optimizing life at its smallest scale.
Beautifully said. Thanks, Basudha. This was for the books.
It is always my pleasure, Dr. Khurana. Thank you so much.
Thank you. Thank you, everyone. Thank you for listening. Bye bye.
Thank you.